The revolutions come from giving up on some fixed idea that everyone holds and realizing that actually things could work in a different way. And that's happened again and again and again.
So you recently published a book, Is Earth Exceptional?
Yes.
What would be your one sentence answer to that question?
Well, my one sentence answer is that that's an interesting question, right? and we don't know. It's just so exciting that like half of astronomy these days is focused on finding exoplanets, trying to look at them in more and more detail, ultimately trying to get some signal that there might be signs of life on other planets. And that's really what we need to do, right, in order to know whether Earth is unique, incredibly rare, or we know that there must be millions of Earth-like planets orbiting sun-like stars just in our galaxy, let alone the whole universe, right? I think we all want to know, is there life out there, or is Earth somehow so special or so lucky that life only got started here? That's the big question.
What would you do if you were given a large sum of financial resources, or let's say even unlimited resources, what we have here, as many people work on that project? What would you do?
Certainly, we could use more money for the sort of small-scale lab experiment. We were very fortunate to have funding from the Simons Foundation over about a 10-year period. And I think that funding really revolutionized the field. It let people do all kinds of exciting new things. And it brought together this confluence of early geology and studies of planet formation in early environments with the laboratory-based chemistry. We need to continue that. We're very lucky to have support from some foundations now, like the Sloan and Moore Foundations, but it's a smaller scale. I mean, there are so many fantastic young people out there starting their own labs, studying these problems. We need more support for them. On the astronomy side, the budgets are 1,000 times bigger.
Astronomical.
Right? They're astronomical, right. I mean, we need millions of dollars, but to do a space telescope, you need billions of dollars. So it's a whole different level of challenge, and the timescales are longer. I would find it very hard to be involved in a space mission, right? That it might take your entire life, your whole career, to see something come to fruition. Like, people worked on tests, for example, for a long, long time before the harvest of the number of exoplanets came through. It's such a huge investment on long time scale. For us, we do experiments where you think of something, give it a try, and we know the answer maybe weeks or months.
Is there a technology missing, like, PCR was like the key to a lot of things that came afterwards in biotech. Is there like a technology, like for example, origin of life experiment on a chip that would allow much faster progress, rather than doing the experiments in like a traditional format, doing them in some kind of, like CellX style, like some kind of mass parallelism, or, some kind of spectroscopy modeling. I don't know, I'm just making stuff up. But the question is, have you thought like what would be like a technology that could take the same people and same resources in a way, but then amplify them the way that some technologies have done for biotech and other fields?
Okay, so I would say two things. So first of all, we are always trying to use the best technology that's available. Mm-hmm. So modern DNA sequencing, for example, that is something that has now become integral to the experiments that we're doing. We can look at RNA copying chemistry experiments on a huge scale. What we used to do is we would take one RNA and see how well does our copying work, right? Now we can take basically all possible template sequences and do the same experiments and see how different sequences get copied, where the errors come in, what context the errors come in. So this new technology is extremely helpful. We're always trying to use the latest advances in microscopy, for example, to look at our protocells, understand how they're growing, how they're dividing. We definitely try to use the best technology that's available. On the other hand, this idea of parallelizing things always comes up. What if you could just try millions of things, right? And see. My answer to that is that what's limiting is actually the ideas, the concepts. And you can't parallelize that, right? You just have to have people thinking about the problem. I think that's where our real roadblocks have always been.
Perhaps maybe LLMs or AI can contribute to generating those ideas which would go into the labs.
Well, maybe. We'll see.
There's one skeptic here, at least, of that. I don't know.
But it's such an interesting answer because that is literally also what Sirac and Soller, whom we have recently interviewed, who made the first design of a quantum computer,
Yeah.
they said the same thing. It's idea limited.
It's the ideas.
Yes.
Yeah, I definitely feel like that. And the reason is, if we look back at the advances in the field of origins of life, this is something I tried to emphasize in my book, that the revolutions come from giving up on some fixed idea that everyone holds and realizing that actually things could work in a different way. And that's happened again and again and again. It's the advances in how you think about the problem that really lead to new development.
So how would you inspire younger minds to come to the field? Or maybe you have something else to tell them. What should they do?
Well, there are a lot of important questions in the field. So, we really need young people to come into the field. But I also have to caution them that it's a difficult field. You really have to be thinking out of the box. And it's also difficult for just funding and just being actually able to do this because it's curiosity driven, right? We just are interested in it because it's such a fundamental question. How did we get here? But most funding agencies want to see something useful coming out of it. And
This is actually one thing I was going to ask about is let's assume that again, we're getting better technology. People have been thinking about this for a long time. There's, more clues. In 10 years, 30 years, whatever the number is, there's some kind of really meaningful breakthrough where it's kind of solved. So we've either made life synthetically, maybe it's not the life that happened originally, or we figured out how it happened, whatever the case may be. What does that future look like? It's always hard to imagine this stuff, but I actually think that probably there would be a lot of applications. So I wonder what that future would look like. What would people be working on as a derivative of that? Basically, once you can make self-replicating nanotechnology, basically, what does that mean?
So in terms of the evolution of life, I think where it's heading is that the next steps would be developing an understanding of, how did metabolism evolve? How did all this chemistry that used to go on out in more or less extreme conditions, volcanic regions and stuff like that, UV catalyzed, how did that turn into something that happens under very mild, gentle conditions inside cells? Thousands of reactions going on all the time in every cell, in every bacterial cell, every cell in our bodies. How did that develop? So that's something a few people were just starting to think about, some initial steps there. Also the process of translation, right? How do you have the genetic code giving us, proteins that can do all kinds of amazing things? How did that develop? That's something we've started to work on, little bits and pieces of that, but there's so much to do in understanding that. Membrane transport machinery, which is, fundamental to everything, everything in neuroscience is about transporting ions across membranes, right? How did that all develop? I think once we solved the earlier questions of how did the first cells get going, then we can start to think more rationally about how they got more complex, how they started to adapt to their environment, explore different environments, and evolve all this internal machinery. So that's kind of where I see the field going.
Is your guess that once there's a self-replicating system, that things can go quickly in the sense that it'll be possible to kind of speed up the evolution by doing artificial selection in the lab or things like that and get a lot of interesting results that way versus is there a good understanding of how long that took on Earth?
No.
Was it a year, a million years, a billion years? Is there a rough?
There's an almost billion-year window of uncertainty there. So obviously the early Earth had to cool down to the point where you had liquid water. And so that's maybe 4.4 billion years ago, maybe 4.3 roughly. The earliest incontrovertible signs of life in terms of microbial fossils are in the range of maybe 3.5 to 3.7 billion years ago. So there's a huge window there.
Are the fossils that we found already roughly like normal bacteria?
Yeah, these are basically stromatolite fossils, so they're probably similar to stromatolites you
that are
see
around today.
Yeah.
So we're missing everything
Everything
in
in
the whole
between.
stack
Right.
before that.
Yes.
Wow.
And we have billions of years to figure this out. Regarding the
Maybe you do. I only have a few years left to figure
well,
this
if we
out.
fix the aging problems now with
Yeah.
telomeres.